helpful discussions. Thank you.
Thank you, Jeremy. Do we have questions from the audience? Maybe I can get things started. So if I understand it right, this picture of you break the connection between the previously polymerized monomer when you then add another monomer to the chain leads to this kind of two landscape picture of incorrect and correct. What happens if you say, well, actually, the bonding interaction persists for a small number of monomers afterwards? Have you thought at all qualitatively about how that changes the picture?
Yes, I think that the effect of roughness would still sort of you'd still sort of get the same effect of roughness. You'd probably get sort of bigger, sort of similar to the equilibrium case. You'd probably get bigger barriers and either side, but I think you would still get that effect of roughness.
Other questions for Jeremy? Maybe I'll go with one more. So the picture you have where the increasing roughness of a given landscape makes it easier to, or makes you more likely to jump to the other landscape that you have a choice about. I guess I understand that intuitively, but also if the roughness consists of drawing that landscape further away from the competing landscape, it almost feels like I may slow my dynamics on one given landscape, but doesn't that make me more likely to stay on that given landscape if I've got deeper wells that I'm getting
Yes,
stuck?
that's why when we measure the effects of heterogeneity, it's relative to that independent case, right? So that independent case would consider sort of almost looking at those two landscapes independently. So that sort of only considers the effect of heterogeneity without considering the energy of each of those two.
Okay.
Yeah.
Okay, got it. That makes sense. Yep. Okay, I think we have time for one more quick question if anyone is moved. Okay, if we can have the next speaker set up. Evan Peepoff.
Yes, hello. All right, can you all see my screen okay?
Yes. Take it away, the floor is yours.
Okay, excellent, thank you. All right, so yeah, thank you to the organizers and all of the attendees. My name is Evan Peepoff. I'm in Professor Jinshu Child's group at MIT. And the title of this talk is On the First Passage Time Fluctuation Theorem in Complex Biomolecular Networks. So I'm going to start by introducing a result most of you are probably familiar with, the well-known detailed steady-state fluctuation theorem, perhaps the most famous result in non-equilibrium statistical mechanics. It's, of course, there are many variations. It's inspired many applications, but basically the probability distribution for producing a certain amount of entropy within a given time window t is related to the probability of producing the opposite amount of entropy in the same time window. That ratio is exponentially related to the amount of entropy produced. Now, one can write a conjugate relationship based upon time distributions, so the time necessary to produce a certain amount of entropy. And so this was done a few years back by Neary and Roldan in some of these papers down here. It's a very nice result. The more generalized result is based upon the martingale theory, but basically the waiting time probability distribution for producing a certain amount of entropy is related to the probability distribution for producing the opposite amount of entropy. The ratio is exponentially related to the amount of entropy. And the time dependence divides out on the right-hand side these are unnormalized probability distributions. So the normalized distributions are equal. And so I'll explain what that means in a moment. So here's just an example. This is a pretty simple, straightforward example to show this. This is like an enzymatic chain reaction. A is like a substrate, B is like a product. And so the total entropy for a basic like first passage trajectory is going to be based upon the chemical potential difference. And that's going to imply that the normalized distributions, the forward and backward times are equal. And not just the mean times, but they're equal at all orders. So that may seem somewhat counterintuitive, but it actually makes sense if you think about it. The probabilities, presumably, between the forward and backward processes are different, but the times are the same. So even though you expect, presumably, more forward events to occur, when a backward event does occur, the time it takes is the same. You can sort of see this illustrated here. It's sort of like a generalization of the two-channel decay time problem. So if you have some state, it undergoes Poissonian decay, it splits into two channels. The rates will be different, the probabilities will be different, but the times will be the same. And if you had a non-Poissonian decay where you had multiple intermediate states, the symmetry is preserved in such a way where the probabilities will be different, but the times will actually be the same. So it's a really kind of neat result. So I had spent some time thinking a lot about complex networks where you have hidden currents and cooperative processes and multiple competing processes. One could be based upon thermal changes, one could be driven, something like this. And I was thinking about how these things could apply to such a network because the statistics are much more complicated. You have the cooperative effects, you have non-renewal effects in many cases where the basic renewal theorem, which you may be familiar with, breaks down. And so I was wondering if we could write similar relations or corrections to those relations or something else. And so the previous result, it assumes that all first passage trajectories produce the same amount of entropy, like by definition. But if you have like some manifold of states, say this is like some free enzyme changing between conformations, some intermediate set of states, enzyme substrate complex changing between conformations, and then you have some product release. So it's cyclic about these states here. You can start and end in different underlying states. So your first passage trajectories can produce different amounts of entropy, where the first passage time is based upon the process completion time to form product, and not necessarily based upon producing one given amount of entropy. And so I'm wondering for this type of, because these distributions can be measured, these forward and backward waiting time distributions. You can't resolve the difference between the conformations, but you can detect product release. And so when you have this kind of hidden process, can you write something like the first passage time fluctuation theorem or some correction to it or something like this? And so that's because these conformational changes are ubiquitous, throughout biophysics, and they occur on timescales commensurate to the active process. And while it's fashionable to neglect them, a proper analysis should often take them into account, especially when you see these cooperative behaviors and you have say chemomechanical coupled motors, and you really have to take into account a hidden process as well as some observable process. And so that's sort of what we look at here. What can we write such a relation? Can we quantify the deviation? Does it contain information about your underlying mechanism that could serve as a signature of something? That's what we look at. So I'm going to start with the minimal model. I'll back up to more complex models in a moment, but this is just basically the enzyme reaction with, it's the minimal model where you can get cooperative behaviors in a fully reversible thermodynamically consistent scheme. So in your free enzyme state, you have some conformational changes, but not in your bound state. It's technically a renewal process, even though it's cooperative. But it's an unusual renewal process because you can start in multiple states, driven by the changing chemical potential. You have a substrate loop and a product loop where we have some local detail balance constraints. But even when we satisfy local detail balance in those two loops, we can still get a hidden current, a conformational current. That's what drives the cooperativity. Presumably, these are based upon thermal changes, so this is non-dissipative. though much of our analysis applies to the case where you do have a hidden dissipative current. But because the math and the kinetics is very hard, we want to start with this simple example first. So yeah, that's our current. And what we find is that you can basically write a correction to this relation that's going to be proportional to the hidden current for this single loop. And this relation is powerful because it works for any single loop or any scheme where you have many loops and all the hidden currents drop out except one. And so you can use this to then show that when all the hidden currents drop out, you'll recover this relation, which is kind of surprising because you'll still have like first passage trajectories that start and end in different states, but they'll all produce the same amount of entropy. And why that is the subject of further analysis. I mean, I have a slide, but I may not have time for it. And that, of course, then implies the Haldane relation. We can test this experimentally, and when it's broken, that serves as a signature of hidden detail balance breaking. So even though you can't measure the hidden current, you can measure these observable processes and infer something about what's going on in your hidden process. Um, and so these results actually apply to a wide variety of complex networks. You know, in addition to the three-state model, you can do the four-state, and you can do lots of complex schemes where you have just manifolds with many different conformational changes. Um, Proving that is tricky, but we have some tricks and been able to establish that. When we integrate this to look at these probabilities are based upon the normalization constants. And what this serves as a forward-backward directionality. And so this is based upon the cooperative kinetics, and then this is based upon thermodynamics. They each kind of serve as a directionality. And we find that that this kinetic branching ratio is actually bounded by the entropy associated with the first passage work. And so this serves essentially like a friction coefficient with the correction to this integrated fluctuation theorem being proportional to the J squared. So it's kind of like a non-dissipative friction-like effect that's diminishing the branching ratio. And this is surprising because you can have positive cooperativity where you have like a speed up enhancement over the Michaelis rate or what you would get without the hidden dynamics. But even when you have that, your branching ratio will still be less than what you would get when the bound is saturated, where the hidden current vanishes. And this seems to be a very general feature. I should also note that like proving this requires a lot of pain, but very pleased that the end result is actually quite clean. And we can say something kind of interesting about these very like complex networks. Um, and then lastly, I guess I'm just about out of time, but I just want to note that we have a stochastic thermodynamic analysis that complements this, where we say, well, what is the meaning of like all the first passage trajectories producing the same amount of entropy? Why does that work? If we do a basic entropy balance along our trajectories, and our first passage work will be determined by like, you know, the affinity or set of affinities, the chemical potential, the force, whatever it may be. We need these two terms to match up in order to make total entropy just based upon this first passage work. So this is a change in stochastic entropy that's going to be related to the populations. It's like a change in surprisal, like what goes into a Shannon entropy. And this is a change in free energy of a machine. It's or the the enzyme or whatever. It's basically like an energy, but accounting for intrinsic entropy. And so one's based upon the populations, one's based upon the rates. They'll pair up in such a way that all the currents drop out, which shows thermodynamically what this means to have no hidden current. It means these terms are matching the change in stochastic entropy and the change in free energy of your conformational states in your machine. And there's a little more to it, but that's pretty much all I have for now. But yeah, any questions?
Okay, thanks, Evan.
Yeah.
Yeah, questions from the audience. We got Rucheng first.
Hi, very interesting talk. So, is your result saying that when we are considering first passage statistics between coarse grain states, the original fluctuation theorem will not hold anymore? Is my understanding correct?
Yes. It's because of we can't resolve the difference like within the individual states because of the course grading process. But when the currents drop out, then it's recovered.
I see. And about your inequality, it it gives me a feeling that it's like coarse grain entropy production is smaller than fine grain entropy production. Is that something related? No.
Could you repeat that? Coarse grain entropy production is what? I didn't hear the second part you said.
This P, so this P plus over P minus,
Yes.
maybe this quantity is related to a coarse grain entropy production. Is that related to?
So it's not. Oh, I see what you're saying. Um,
And give me this
no,
feeling about
I think really the way to think about it is that one is kind of based upon kinetics and one is based upon thermodynamics. But the thermodynamics for the coarse grain states, that's a slightly different component. We actually have something related to that in one slide here. But it really has more to do with like, yeah, so I mean, you can have like an entropy component for your coarse grain states, and it can go into like how you think about microscopic reversibility between your coarse grain states, which like breaks down but can be restored. But so it's yeah, you're thinking along the right lines. Though it's slightly different though what I was describing there.
I
Yeah,
see. And
good
about the
question.
effective friction coefficient, is
Oh,
that a measurable quantity? The effective friction coefficient. Is that measurable?
let's see. Ah, no, that's a very good question. I think you can measure it with some tricks. It's not easily measurable. Yeah, I think that you can infer it with some tricks, yes. But it's not
So
direct.
okay, so this is the first equation is a definition of this effective friction coefficient, or it has another expression.
Oh, this is just so if you take
Do you
this
have
general
a
expression and you look at in Laplace space and you look at, z is 0. So it's like an integrated first passage time
okay,
fluctuation theorem. It's just what you'd get when you have the J. When you have no J, you just recover this, which is like the integrated version of it's just with P pluses instead of the distributions, basically. So this is a bunch of rate constants. Actually, I can show you what it is. It's this,
oh, I see. Thank
which
you.
is
Thank you.
amazing because these distributions are huge quantities. So the fact that we can get this simple expression is kind of nice. But that's what it looks like. Yeah. And so yeah, it's just a combination of rate constants, this effective like friction coefficient. And that's just sort of an interpretation.
Thank you very
Okay.
much.
Yeah, sure.
Yeah, I think we need to wrap up our discussion now, but let's thank Evan again for an interesting talk. Evan, if you can stop sharing, and then Timo, if you want to share your slides. So we are transitioning now from the lightning talks to 2 invited talks to wrap up today's session. And so our next speaker will be Timo Betz. So Timo, please take it away.
Thanks a lot. Can you see my screen? That works? Cool. Awesome. Yeah, thanks a lot, David. Thanks a lot to the organizers for giving in this slightly theory-biased conference or workshop an experimentalist the chance to talk about his or like our work. But of course, I mean, this is not only my work. This was to a large part here worked together or worked out by Gabriel Knotz here. This is an amazing PhD student in the group of Matthias Krüger, who is a theoretician here in Göttingen. And the experiments I show you are to a large part have been conducted by Till Münker. So what we care about in the lab is really trying to figure out what's going on in the world of biology when you look at projects or processes like this. This is a cell division, what you see here, and there's lots of complex stuff going on. And really, I don't want to go into the biology here. I just want to like share with you a little bit the problems that we have and the methods or the ideas that we tried to come up in the past years to solve them. And among these, there is one nice new quantity that I would like to pitch to you. So the problem really is kind of obvious here. I hope the complexity of the system is basically killing you. So we have 40,000 different genes in there. They all work together or more or less in a coordinated fashion to generate all of these complex behaviors. And using the tools of statistical mechanics is extremely powerful, but here we have also activity. So we have this highly non-equilibrium situations, which makes it very hard to apply these. So our aims in the lab are basically first to figure out if a system is out of equilibrium, because it is not guaranteed. It could be that you have substructures inside cells or certain time scales where it is totally okay to apply equilibrium physics, and then we would have this powerful toolbox, right? But we would like to have some easy tests to figure out, is the system in equilibrium? And then the other thing that we would like to do, or our aim is to quantify them also this extent. So it's sometimes sufficient to know if you're not in equilibrium, but it would be great to know how far. And of course, the final goal of many of the work that we do in the lab is to understand the mechanical properties, because at the end of the day, a lot of the processes that we see here on the left turn into motion or change the viscoelastic properties of the cells. So one of the solutions that we are actually looking into here is, besides using more classical or let's say straightforward ways of testing for an equilibrium, would be to find a new observable that we call mean back relaxation. And it turns out to be a very powerful way to actually detect the degree of broken detailed balance in these systems. And all you need is just a trajectory, which is very convenient from an experimental point of view. All right, so I hope I can motivate you to listen to an experimentalist now. And you will see first the main question that we look at, and that is this one here. Looking at such a cell, right? It's an oocyte of a mouse. You see a lot of dynamics going on, and we know that the system is alive. So you get this intuition, oh, well, that's probably like non-equilibrium. But I mean, just looking at our good old Brownian motion, it turns out that actually the statistics of these particles here moving around is quite close to what you would have here. So a mean square displacement of these particles basically shows you a diffusion process. Also, we know the system is out of equilibrium. It's actually quite hard to pin it down. And so the first question that I will talk about in the next few minutes is, what are ways to figuring out if the system is not Brownian only, but also active? And the first part of the, let's say, more classical thing we do in the lab is really checking the fluctuation dissipation theorem. And this audience, I don't have to explain what that is. In simple words, we just try to measure on one side the fluctuations here by a power spectral density, and on the other side, we try to find an independent measure of the response function here, the dissipative response function. And we do this using optical tweezers. In a nutshell, that's actually quite a convenient system. You focus a laser down, for example, here on a particle inside a cell, and then you collect the light that is going through here, so the scattered and the unscattered light, and you collect that on a detector. And then you can learn here both things. You can learn about the force you apply on this particle, so you can move that particle around, and you can also turn the laser almost off so that there's just sufficient light there to just get the scattering, and that tells you about the fluctuations of the particle. And what such a measurement does is just showing here. The first experiment here would be just measuring the fluctuations. So we have a phagocytosed particle here. We turn the laser almost off, so there's just this scattering happening, and then we see a free fluctuating particle here. And out of this, we can extract very precisely the trajectory. We do a Fourier transform, square it, and then we get here the power spectrum. And now we apply naively the fluctuation dissipation theorem, and we get a prediction for the dissipative response function. Okay, that's this. Now the big question is, of course, to which extent is this maybe or maybe not in thermodynamic equilibrium? So is this equation actually valid in this situation? And to test this, we can do a second experiment. And Ivan, if you have been in the first part of this afternoon, already talked about this. There is a way where you actually apply a known force to your particle and directly measure the response function. So this was already introduced a bit by Ivan. So what we have here is we apply an oscillative force and we measure the position. So in blue here is the force that we apply, and you can see in green there's a smart delay of the response of your particle. And the definition of the response function, if you want here, is just the convolution, or just is a convolution between the response and the force applied. So if we just go now to the Fourier transform of that, we can very easily get the response function. And if we plot now in there the dissipative part of this, we can actually have a measure like this. Okay, and then we do this for many frequencies, and then we get curve like this. So what such a curve tells you, and this is now just some data from an MDCK cell out of an epithelial cell layer. What you can see here, we have two branches. The upper branch basically is the predicted response, assuming fluctuation dissipation theorem is true, right? So this is assuming that that equation is true. We can see that measuring the response independently gives us smaller values. That just means that the reason for the fluctuations is not only Brownian motion, but there is some activity involved. And what we do now is, or what we would like to do now, of course, is quantify this activity. And we'd also like to know if there are certain regions or certain regimes where the activity is not important. You can already see it quite nicely in this plot. If we go to higher frequencies, and that depends a bit on the cell type, right? Typically starting at a couple of 10-ish hertz, like, or sometimes later here in this one at a couple of 100 hertz, the system shows that fluctuation dissipation theorem can be valid. This is important because if you look at short time scales, right, you can actually use it. And I think this is a very nice take-home message. If you actually want to look at longer time scales, or you want to average over longer times, then you really have to be careful because activity really kicks in. And you can quantify this now very simply by assuming that our fluctuations are not only driven by KBT, but we just introduce something we call effective energy. Of course, you have to take this effective energy with a grain of salt, because it is not necessarily really just the energy applied to the system. It kind of assumes, or it's a placeholder for KBT in a sense that explains, or that connects still the dissipations and the response. So if we kind of like do this, the nice thing is that this effective energy, which is a measure of non-equilibrium, right? This can be directly accessed experimentally. It's not an easy measure, but it's possible. Now, let's take a look at this effective energy. What can we see there? So if we just plot this now for two different situations. One is a normal interface cell. Interface just means between two cell divisions. It's more or less in a steady state situation. And the second, the red curve here, would be a cell where we have depolymerized the actin cytoskeleton and the microtubule cytoskeleton. In simple words, what you have inside cells are motor proteins, and these motor proteins kind of like drag particles around. Now, these motor proteins require a substrate to walk on. And what we do here is we don't kill the cell, right? Because if we kill the cell falls apart and we cannot measure anymore. We just remove the substrate for the motors, okay? And that's very convenient because the cell is still alive, right? We can also recover this, but we see that here, this energy here, what we do here is we actually subtract on top KBT. So that's why we call this active energy now. But we can see very nicely that the same cell, after removing the motor activity, basically this active energy collapses. It's not fully dead. You have to keep in mind the cell still has pumps, you know, there might still be polymerization of other proteins going on, but it's really nicely collapsing. So that's a nice control measurement. Another thing that's quite striking, and that is something we will need maybe later, is that we can see power laws here. So this is not an exponential decay that we see here in the frequency range, but we can very nicely associate power laws to this. And typically we find power law exponents of -1. So in simple words, if you think that this active energy process is something like a noise, like an additional noise process, it shows this beautiful 1/f dependence. And that really comes directly out of the measurements, right? There's nothing imposed here. All right, so take-home message here would be that there is a way to quantify activity in these cells, right? By comparing fluctuations with the dissipation process. This activity turns out to be more important in lower frequencies. And if you get quick enough, then the system seems to go very nicely to a KBT region. And we can nicely see that there seems to be a phenomenological power law dependence here. We get one thing more, just to be complete. Since we got this response function, right, we can also take not only the dissipative part, we can also take the real part, and that gives us access to the full mechanics of it. So, right? So that's how that would look like here. This is a frequency-dependent modulus, like shear and the dissipation modulus. We can model this by a power law, and if you care about this, we could talk about it later. So in simple words, our aim, right, that I showed in the beginning is fulfilled. We can show that the system is out of equilibrium. We know to which extent it's out of equilibrium, and we can get the mechanics. But this was a very tedious, actually, measurement. And the idea or the hope when I came to Göttingen was that I can kind of like try to use all the smart theoreticians there to figure out a better way. Because experimentally, what we have to do at the moment is we have to do 2 experiments. That means once we just observe the fluctuations, we just watch the system. That is easy to do. And the other one is you have to do a very well-defined perturbation. And doing such perturbations is experimentally quite challenging. So wouldn't it be cool if we can get rid of this part? Wouldn't it be nice if you just watch the system and recover like passive microbiology? And the problem is, of course, passive microbiology is not necessarily working in active systems. So is there a way to figure out that the system is breaking detailed balance and is active? And we're thinking back and forth, and then after some time, this was really very beautiful collaboration with Matthias and Gabriel. So we meet once a week, and we just discuss these things. And then we had many ideas that led nowhere. And then at some point, we were thinking about this rather simple idea. We were asking ourselves, to which extent is actually an active measurement different than a passive measurement? And if you think about a simple active measurement, which would be here in the upper left, just having a particle, dragging it somewhere, and then letting it relax, right? That would be the most simple active experiment I can think of. You can also take yourself the hand and think like, wait, let's just look at the spontaneous fluctuation and wait until by chance the particle was also going here from a, let's say, this right situation to the left position, right? Okay, now listening to Onsager, right, who tells us that the average regression of fluctuations will obey the same laws as the corresponding macroscopic irreversible process, right? In simple words, if you look at the recovery here, like this relaxation, it doesn't matter if I pushed it there or if I just watch it afterwards, it has been fluctuated there. It should be the same thing. So we basically just look or use that idea. And then we came up with a simple protocol. So the idea is really simple. We collect data, we collect free fluctuations, and then we kind of like pick out the trajectory, the part of the trajectory that fulfills 2 simple conditions. One, at a certain time t, it's in a certain position x naught. In our example, let's say it's just here to the left, right? And we also require that in its immediate history, it had been at this other position here. So in simple words, we just pick up all the situations where the particle has traveled in a certain time from here to here. And that can also happen by chance. Of course, the statistics goes down the bin, but there is a way to actually recover this. So how does that look like? Well, this is a simulation here of a particle in a harmonic trap, and we zoom in, and that would be just one of these trajectory that kind of like fulfills that condition. It's here in a certain moment, right? And in the past, it had been here. Right? This point, for example, if you go back and up here, would not fulfill the condition. Now we just put this to the side, and then we do it again, and we do it again, and we do it again, and we do it a couple of thousand times, and suddenly you come up with like trajectories like these. There's something that I need to tell you. What I did here is I also normalized this trajectory by the distance d it has traveled. And this is an important trick because by this we can recover statistic. Because